Okay, this is part 3 of the nervous system. I'm going to jump right into it. We have a lot to unpack. So, parts of the neuron. We took care of histology in the previous one. So, we have the soma, or the perikaryon, is the cell body. So basically, and you're going to see this in a little bit. And then there are going to be all of these extensions, right? These are going to be dendrites, and you guys know I can't draw. And then this is going to be one axon. Here's going to be a nucleus, some rough ER that we call Nissl bodies. So this part right here, that is the soma. And from the soma is where these neuronal processes are going to be. This is underlined, just means that's the next thing that we're going to talk about. It has no centrioles in here, right? It's amitotic. It can't go through the cell cycle. And the rough ER are called Nissl bodies. There's going to be this triangular region where axons arise. This region right here is called an axon hillock. And I'll show you this axon hillock again in an image. So the processes of neurons, there are going to be two types, axons and dendrites. However, in the CNS, they're called tracts. So when you take a look at a cross-section of a spinal cord, there are going to be tracts in there. And when you're in the PNS, the peripheral nervous system, they're just referred to as nerves. So the dendrites are short, tapering, and these are the receptive regions of the neuron. And signals are conveyed as graded potentials, not action potentials. Excuse me. And I will cover the difference between these two. But it's really important to understand that the dendrites are the receptive regions, right? God, I really can't draw. So here are the dendrites. Here's my axon hillock, and then here's my axon. So these are the receptive regions. They're going to receive a signal from some sort of stimuli, and graded potentials, if they are strong enough, they will then convey an action potential down the axon. And again, I'm going to cover that in greater detail. The axons now, they're the same diameter. They arise from the hillock. There's usually one unbranched axon per neuron. If branches are present, this isn't too clear here, but if branches are present, they're called axon collaterals. And the axonal terminal is the branched terminus of an axon. And we already know that, right? Here is this. And here are all of these vesicles containing neurotransmitters, right? So that's the axonal terminal. And here we can see this here. So all of this right here, this is the soma, kind of what you would think of as the cell. Here's the axon hillock. And then here's the axon, and here are the dendrites. Graded potentials, action potential, and axonal terminals, okay? So the function of these axons then is to generate and transmit action potentials. And they secrete neurotransmitters from the axonal terminals. That's a review. Movement along the axon occurs in two ways. Anterograde is towards the axonal terminal. Retrograde is away from the axonal terminal towards the soma. The myelin sheath, we briefly covered this. It protects the axon and insulates the fibers. The other thing, myelinated fibers, they dramatically increase the speed of the propagation of that action potential. And we're going to talk about why later. So how does this myelin sheath form? We know that it's formed by a Schwann cell in the PNS, right? And it's an oligodendrocyte in the CNS. Well, basically what's going to happen, you might want to pause it here. I'm just going to jump ahead to the next. So this Schwann cell is going to keep coiling around the axon. And as it does, it's going to squeeze. So you're going to have all of these layers of plasma membrane. The neurolemma is this outermost layer. It still has a little bit of cytoplasm, and that's where the nucleus will be as well. Right? It'll be part of that neurolemma. So that neurolemma is just the outermost layer of the myelin sheath, or of the Schwann cell. Now, what we see is that there are going to be gaps between Schwann cells, that myelin sheath, and they're called nodes of Ranvier. And remember what axon collaterals are. They are branches. So if a branch is going to happen, kind of makes sense that it's going to happen at these nodes of Ranvier. See, these nodes of Ranvier are these gaps. So the myelin sheath, one Schwann cell does not completely touch another one. There's going to be this gap, and that's a node of Ranvier. And if an axon collateral is going to happen, that's where it would occur. And then basically, unmyelinated axons, we have a Schwann cell surrounds the nerve fiber, but coiling does not take place. And then we have CNS, axons of the CNS. We have myelinated and unmyelinated. Again, they are formed by oligodendrocytes. There are nodes of Ranvier, but they're widely spaced. There is no neurolemma. And basically the difference between white and gray matter are myelinated. Gray matter consists of unmyelinated axons and the soma. It's a lot to unpack, but you're just going to have to kind of review this a few times to get this down. Next part, we're going to start talking about graded potentials.